In this lecture, we will cover the second set of material on Chapter 12, as we are studying Neuropeptides II, neuropeptide function. And we pick up with this slide reminding us that we are beginning a discussion of the electrophysiological responses of neurons to neuropeptides. As we had stated, there are a few examples, such as substance P and neuropeptide Y, that regulate the firing rate of neurons in the spinal cord and the sympathetic ganglia, and vasopressin can have similar effects on hippocampal neurons. So we had viewed this slide to remind ourselves of how we can have recording electrodes either outside of or inside of neurons to pick up the membrane potential. With a reference electrode here and an amplifier, then we'd view the signal. And stimulating electrodes that could be either at the soma, on the dendrites, or on afferents inputs that would be coming to this so that we could activate the axons and then get the release of transmitters. So we would be looking at natural synaptic effects on these neurons. So a variety of approaches, as well as similar type of glass capillary pipette or other cannula type materials could be used to apply substances such as neuropeptides for our discussions or neurotransmitters at the same time that you're electrically stimulating or related to each other, peptide before stimulating, and so on and so forth. The effects that we are considering are changes in the membrane potential, going from resting membrane potential, depolarizing closer to threshold at different magnitudes. So over time, we see that as you increase the membrane potential closer to threshold, then the neuron is capable of firing more action potentials based upon synaptic input or other chemicals applied, or electrical stimulation of synaptic inputs. So these would be millivolts on the y-axis and time in milliseconds on the x-axis, as we see in this slide showing the response of the neuron membrane potential to increasing intensities of stimulation. We'll see another slide where intensity would stay the same, but the frequency of the electrical stimulus pulse can be altered. So you can alter stimulus intensity or frequency of the stimulus firing, and both will produce a similar type of a dose-response reaction if you are wanting to consider it that way. So again, as the membrane potential approaches and exceeds the threshold, the stimulus itself will cause the neuron to fire action potentials, and the higher above threshold you go with the stronger intensity stimulus. You can see that the stimulus time duration is approximately the same on each of these cases. The number of action potentials over that stimulus duration can increase. So these are some of the basic concepts that we need to have in mind as we proceed to study the electrophysiological effects of some of the peptides. And we recall that although this is time in milliseconds here, each of these represents an action potential. So if we take this time scale that must be hundreds of milliseconds and stretch it out so that we could see individual milliseconds such as this, then each one of those action potentials should look like this. And we remind ourselves that whatever the stimulus is to depolarize the membrane at reaching threshold, the voltage-gated sodium channels open, the sodium influx will occur, so there will be an inward current of sodium over a period of time. Then the sodium channels are inactivated and closed, and they no longer contribute to the current. So now the potassium channels will have finally opened, as described in the prior lecture. Excuse me. And there will be an outward an outward directed current due to the potassium. It'll reduce the positivity inside, obviously, and the inside becomes more negative, in other words, repolarization. There is an overshoot because it takes certain conditions like reversing the polarity, becoming hyperpolarized, to close the potassium channels. Potassium channels contributing to the repolarization can be targeted by peptides, as we will see, and calcium channels can be affected that will impact the shape of an action potential as well. So we want to remind ourselves of the permeability, the ion permeability, in this case shown on the right-hand scale for the potassium, and the sodium curves shown here. And we have the action potential, of course, with the membrane potential on the y-axis to the left, and milliseconds. So there is a tremendous increase in sodium permeability that decreases as the sodium channels have closed, and it corresponds with the rapid rising phase and the peak of the action potential. Decreasing sodium permeability and peaking potassium permeability, of course, correspond to the rapid repolarization and contribute to the hyperpolarization. So if we're going to change the shape of the action potential, we are not likely to impact the sodium channels. The more common effects that we will see are impacts on the potassium channels, so the shape of the action potential from the highest point, the peak, onward to recovery of resting membrane potential are most likely to be affected. Peptides might contribute to this as co-transmitters under conditions where the frequency of the stimulation has increased. So you see hertz in frequency of stimulation for these events being shown here along the x-axis, and the x-axis is actually in time, and then the y-axis would be relative extracellular levels of the transmitter or peptide co-transmitter. So lower frequencies are going to generate fewer action potentials. Each action potential is going to cause the exocytotic release of a certain number of transmitter molecules, because there's a certain number in each vesicle. Each action potential event brings about a certain number of vesicles fusing to the presynaptic membrane, so the quantal release, as you recall. But which population of vesicles moves to the membrane and docks and fuses to dump its contents differs depending upon the frequency. So a single event, a single electrical stimulus just produces one quantal or one collective quantal release. We don't know if this is just one action potential or 10 action potentials, but anyway, a standard amount of transmitter is dumped into the extracellular fluid compartment, the synapse. With the low frequency, so multiple stimuli, of course, that is increased and sustained over a period of time. As well, some of the peptide-containing vesicles will also fuse and dump. At the higher or highest frequency, in this case, then we can see greater exocytotic release of the transmitter itself, as well as of the peptide, and the peptide release is sustained very long after termination of the stimulus that's shown here. So my proposal to you is that you begin to appreciate the presence of peptides in the synapses along with the primary transmitters, and that even though this illustration kind of shows us a change in the shape of an action potential, it actually is not showing that. If we were looking at one synapse, these chemical presences within the synapse could not work over this temporal period to alter the action potential, because this is the primary transmitter thought to produce a rapid effect. This is the peptide that's going to act only through metabotropic receptors, causing a more delayed change in the target cell. So maybe after an event like this, the subsequent impulses coming in, if they were delayed subsequent to this battery of stimuli here, dumping these transmitters, co-transmitters, could possibly be altered because the metabotropic effects of the peptide could, over a period of time, change the property of the membranes so that a subsequent event would be affected. So this is an action potential from contractile cardiac muscle cell in the ventricle, the left ventricle, and it occurs over a 200 millisecond time period. So this would not be enough time for the transmitter and co-transmitter to produce this effect. Of course, these are features of the action potential of the cardiac muscle cell that are independent of any type of external stimulus. These cardiac muscle cells have an autorhythmicity, making the point that different cell populations, cells with excitable membranes, have different populations of ion channels in their membranes, and it's that population of ion channels in the membranes of the cell that gives its characteristic excitability pattern. So actually the sodium channels are different. They automatically allow sodium in, so it's a spontaneous depolarization. Neurons require some input to open sodium channels, for example, either ligand gated or voltage gated. Then if it reaches threshold, it is an all or none event. The sodium influx would end at about this time. Same thing for cardiac muscle cell. But the potassium channels can't kick in because of the calcium channels being opened here instead, and the intracellular calcium levels have to reach a certain point before the potassium channels complete their opening and contribute to the repolarization event. This produces a very extended absolute refractory period and a very short relative refractory period. This, of course, protects cardiac muscle cells so that you don't have like my speaking muscles are doing right now. You don't want your heart to stop. You want it to contract over the same time period that you have an action potential, so that you don't get any potential summation of events, so that 3 action potentials could occur in this time period and just keep the heart muscle contracted. So there's no summation that can occur in cardiac muscles. So a similar type of thing can happen in neurons, where the neuron cannot reach a peak action potential, or firing rate, of generating action potentials, because no other action potential can occur during this time period. So if this is 200 milliseconds, that's a long period of time if the typical neuronal action potential is about 3 milliseconds. So there's a lot of 3 milliseconds in a 200 millisecond, right? So you reduce the number of action potentials that could be fired by this neuron. And we'll talk about mechanisms by which you can shorten the relative refractory period without affecting the absolute refractory period of the neuronal action potential, and thereby making it more possible to bunch the action potentials all close together and reach a peak firing rate. So we can, by altering action potential shape, we can affect the maximum potential firing rate of the neuron, as well. A short action potential versus a long action potential is going to have different impacts on the amount of calcium that will influx at the axon terminal. When that big giant action potential gets there, it's going to have a different dynamic effect, quantal release, than a short action potential. So these are the kinds of things that we want to take into account. We're thinking about peptide modulation of neuronal function. So what might be some of these channels that we would consider? Well, there, of course, are potassium channels. We see the I sub K leak. This is the leaky one. The I sub Na sodium channel, voltage-gated sodium channel. The I sub Ca, or calcium, the voltage-gated calcium channel. And a voltage-gated potassium channel. It's the delayed rectifier that causes that after hyperpolarization. We'll complete reading this table in just a moment. And the calcium-dependent potassium channel, the I sub K Ca, that we had talked about with the cardiac myocyte. So the first is the leaky potassium, and this produces a relatively high permeability to potassium. P sub K is potassium permeability of the resting cell, so there is constant efflux of potassium, and this makes the membrane negative inside relative to outside. So it's partially blocked by substances such as tetraethyl ammonium, or TEA, and this is largely responsible for the negative resting membrane potential, or V sub rest. So the resting membrane voltage or potential is designated as V sub rest. The voltage-gated sodium channel we saw is going to open during depolarization of the membrane. Is activated, becomes inactivated even if the V sub M, the membrane potential in voltage, volts or millivolts as we know in units, remains depolarized, because it is voltage dependent for opening, time dependent for closing, inactivation, closing, and reactivation sequence. Tetrodotoxin blocks these voltage-gated sodium channels, and this, of course, produces the rising phase of the action potential. Next, the voltage-gated calcium channel. So it's activated by depolarization, but more slowly than the sodium channel. So voltage-dependent for opening and delayed compared to sodium channels, temporal feature for opening. And it's inactivated by function of cytoplasmic calcium concentrations, or the membrane potential itself, the V sub M. So this is a type of autonegative feedback. Voltage-gated calcium channels cause localized increased calcium level. When that reaches a critical level, it triggers the closing of those channels themselves. So kind of an auto feedback type of a thing, an example of a negative feedback system. So verapamil, or D600, cobalt, cadmium, manganese, nickel, and lanthanum are examples of calcium channel blockers. Verapamil is probably the only one on this list that's used clinically. Produces a slow depolarization, allows the calcium to enter the cell where it can act as a second messenger. So there's basically three things that are accomplished through calcium influx. You make the inside more positive, so you can contribute to depolarization. You increase intracellular calcium levels that can have effects like on the cytoskeleton or other molecules, such as docking of the vesicles for exocytosis. Or it can activate intracellular signaling mechanisms, and it indeed itself acts as a second messenger. The voltage-gated potassium channel, the delayed, it's activated by depolarization. So voltage-dependent opening, but more slowly than sodium channel. It's inactivated slowly and not completely if the V sub M remains depolarized. So in a certain type of a preparation, if you can depolarize the membrane and keep it depolarized, the potassium will continue to leak. These channels will stay open because it's voltage dependent for opening and voltage dependent for closing. So there's abilities to use intra or extracellular TEA to block this, and aminopyridines will also block the I sub KV ion channels. So it carries the current that rapidly repolarizes the membrane to terminate the action potential, as you recall. Then finally, the calcium-dependent potassium channels. They are activated by depolarization, so voltage-dependent for opening, plus elevated calcium. So the voltage gating will initiate things, but until the appropriate intracellular calcium level is achieved, you don't get the full opening. It remains open as long as the cytoplasmic calcium is higher than normal, so you have to remove that calcium to terminate this positive effect, and thereby it is calcium dependent for closure. So extracellular TEA will also block these these potassium channels, just as the ones above, and the leaky ones. So all three potassium channels can be blocked with TEA. So this carries current that repolarizes the cell following the action potential based on either sodium or calcium, and that balances the I sub Ca, the voltage-gated calcium channels, thus limiting depolarization by the calcium channels, voltage-gated calcium channels. So the degree to which calcium can depolarize a membrane is limited by the presence of these I sub K Ca, or the calcium-dependent potassium channels. So I think you can see that different populations, different distributions of these voltage-gated ion channels, and especially if they can be targeted by metabotropic type of effects around the membrane of a neuron, dendrites, soma, and then of course much more concentrated on the axon to accomplish what needs to be done there, action potential coupled to exocytosis, but distributed more diffusely throughout the entire membrane of the neuron. You can target these with metabotropic mechanisms, some or all, or maybe there's additions to the list that we don't have here, but we're just trying to envision it, and change their characteristics so you can hyperpolarize the cell by making some less or more leaky, and hyperpolarize and or depolarize, of course, by changing the leakiness or the properties of any of these correspondingly. And thus we come to the figure from the textbook. This is figure 12.4, and we introduce it first reading from page 273 in the textbook. So as shown in this figure 12.4, the neuropeptides can alter the shape of the action potential, shown in the upper panel here, panel A, the frequency of neuronal bursting, and the responsiveness of the cell to stimulation from primary neurotransmitter, shown in the either to electrical stimulation or to transmitter, shown in the other panels. So a change in the shape of the action potential. Here we see the rapid rising phase, the repolarization phase, the after hyperpolarization, and recovery of the resting membrane potential. So it begins to look very much like the action potential within the cardiac myocyte. This indicates an alteration in the time that the ion channels stay open. So if the potassium channels don't open in time, or if sodium or calcium channels stay open during this time, or a combination of those two, you can change the shape of the action potential such that it takes on this characteristic appearance. And it accomplishes the two things we mentioned. You can't fire another action potential anywhere within the absolute refractory period because it's been extended. and it can impact the quantal release features at the axon terminal upon arrival. In panel B, you see changes in frequency and pattern of spontaneous firing rates. For example, somatostatin depresses the firing rate of a single cortical neuron, so that's shown here in panel B. So this is probably an extracellular recording. You see the time scale in minutes, so 1 minute is shown here. So this is a highly compressed temporal scale. Each of these actually represents, it's a histogram, so each of these is representing the frequency of action potentials, so it's an additive type of a thing. We couldn't stretch this out and see each of these as an individual action potential. They're representing the number of action potentials in a certain period of time as we're proceeding. So the higher this the higher these upward deflections, the higher the frequency of the firing rate of the cells that are being recorded. So this is showing the spontaneous firing rate. You begin to bathe the neuron with the somatostatin, and slowly this firing rate decreases and just becomes episodic. So it takes on not only a lower firing rate, but it begins to almost take on a bursting pattern. So the pattern, frequency, and the pattern seem to be affected, and then with washout it is reversible and you recover the normal firing rate. And we will see that somatostatin can have different effects. It decreases the firing rate as shown here, and then in the lower panel it's going to alter the response to another neurotransmitter. But let's take a look at panel C first. So in panel C, what we are looking at excuse me, just trying to find the point in the text, so let's just go ahead and take a look at it. Changing the frequencies of neuronal bursting, so a change in the firing pattern in response to a stimulus. So first we see the membrane potential. So let's in our mind imagine the scales that we would have here. This is membrane potential, so it should be in millivolts. And we do not have a time scale on this particular one, but this would probably be a very fast time scale in milliseconds, certainly not as long. This length of the scale bar would certainly not be minutes. Here it would be milliseconds, individual milliseconds. So without the substance P present, the membrane potential would be at a certain level, resting membrane potential. So positive would be up and negative would be down. And upon stimulus, there's just a single action potential, or no action potential, excuse me, that is generated. So this is the stimulus artifact here, and then here we get the recording, and there is no action potential recorded. We then shift to the experiment where we have a baseline and stimulate there. We get the same response as in the control cell. Add the substance P as shown here, and we see what there is an upward deflection of the membrane potential in millivolts closer to threshold. Not to threshold, so it generates action potentials itself, but then upon stimulus we see that there are multiple action potentials that are fired. So this is what we were saying. The neuropeptides can, through metabotropic mechanisms, alter the ion channel features in the membrane nearby a synapse or in the whole cell itself, and you get the membrane potential V sub m becoming more positive, approaching threshold such that when either an electrical stimulus or synaptic input occurs that's excitatory, it can display characteristic firing patterns, in this case a burst of action potentials. And then in panel D, we are seeing a different type of an effect of somatostatin, wherein it interacts with a neurotransmitter. So the somatostatin can prevent the opening of calcium channels and thus inhibit the spontaneous neuron firing, while others, such as the substance P, close the normally open potassium channels. So if you, and thus increasing the depolarization of the cell until the firing threshold is reached, as we see here. So if you decrease potassium influx, the potassium accumulates on the inside of the membrane, making it more positive relative to outside, so you get the upward deflection. So the substance P is targeting potassium channels, most likely the leaky potassium channels in this case, or whatever potassium channels are contributing to the efflux of potassium, and membrane potential becomes more positive. Here we're affecting the calcium channels with the somatostatin and altering or inhibiting the spontaneous firing of the neuron in the cortex. So this leaky calcium channel can contribute to the high firing rate, the spontaneous firing rate. So as we said, depending upon the population of channels in the membrane of a neuron and the properties of those channels that can be changed through metabotropic or receptor or voltage mechanisms, the neurons will have their own characteristic features as far as spontaneous firing rate, bursting, or responding to input. So let's compare the differences between somatostatin affecting calcium channels and the spontaneous firing rate versus somatostatin affecting the response to another neurotransmitter. I'm sorry to keep you waiting. So what we are seeing here is that the somatostatin, of course, facilitates the firing rate of the cells to the acetylcholine stimulation, even though it depresses the spontaneous firing rate of the same cell as in B. So it can, somatostatin alone can suppress the firing rate by acting on the calcium channels and can increase the response to the neurotransmitters. So here we see the acetylcholine. Again, this is a 1-minute bin looking at the firing rate in response to the acetylcholine causing an increased firing rate. So each time acetylcholine is added, there's an increase, a corresponding increase in the firing rate of the targeted neuron. When somatostatin is added, the firing rate is enhanced. So it seems that somatostatin is going to decrease spontaneous firing of the neuron to a level to where it's almost a bursting type, and then acetylcholine in combination with the somatostatin can then take over the control of the firing pattern, the bursting pattern and frequency of that neuron. So a very different type of a signal when you've got a peptide working in conjunction with the neurotransmitter versus the peptide affecting the neuron directly. The authors provide other examples, of course, of peptides having effects. So we've talked about the potassium channel and the calcium channel. The endogenous opioids also have some interesting effects. Basically, they inhibit neuronal activity by decreasing the calcium influx into neurons, similar to the somatostatin shown in panel B. or by enhancing potassium conductance. So if you enhance potassium conductance, then you will get a hyperpolarization. So it would be the opposite effect of inhibiting or decreasing the efficacy of the potassium channels. So if you decrease the efficacy, less escapes, more accumulates inside, the membrane becomes more positive. It would be the opposite. If you enhanced it, then more potassium leaves, so it becomes more negative inside than outside, so we'd see the completely opposite effect of decreasing the membrane polarity. But there's also some evidence that the opioids can decrease the potassium conductance, similar to what we would see here. So it depends upon the neurons in question and the peptides under study. Now, when the neuropeptides depolarize the postsynaptic cell, that cell, of course, is more likely to show the neural bursting when it is stimulated, as was shown in the panel C. And this can also be demonstrated by the effects of thyrotropin-releasing hormone on respiratory neurons in the nucleus tractus solitarius, or the NTS of the brainstem. So this figure 12.5 is showing us, first in panel A, a cross-section of the brainstem and the medulla. Here's the 12th cranial nucleus that shows the motor control to the tongue. This is the tractus solitarius that's shown here, and around it is the nucleus tractus solitarius. So a lot of sensory information comes in through the vagus nerve into this area. And these neurons have various visceral functions. In this case, they're respiratory neurons. So in panel B, what we're seeing is an unpatterned, and in this case, the x-axis, of course, the y-axis is in millivolts. So this scale bar is showing 40 millivolts for panel B, and the x-axis scale bar is showing 1 second in the x-axis for both of these illustrations, these figures in panel B. So what's happening is under normal conditions, the control conditions, you have the neuron showing spontaneous activity over a period of time, with not a characteristic pattern of bursting, but a characteristic pattern of firing, certainly. When the TRH is added to these neurons, extracellularly and exogenously in this case, then we see that the neuron shifts somehow such that you just get a bursting of the a clustering of the action potential. So a burst with a resting period, a burst with a resting period, and then another burst. This is a very different signal from this neuron than this patterned firing rate as we see above. Now the mechanism by which TRH is doing this is related to events after the action potential, and that's shown in panel C. And we draw our attention again to the axes. Now the upward deflection, the y-axis, is still millivolts, but this similar height is now 10 millivolts. And the x-axis, hopefully I called that the y-axis, sorry. The x-axis is still time, but this is 0.2 seconds or 200 milliseconds. So this is very likely an extracellular recording here, because we're getting 40 millivolt deflections, and here we're getting 10 millivolts, so this is probably an intracellular single cell recording. And at 0 minutes before the TRH is added, we see a characteristic action potential with the after hyperpolarization and recovery of resting membrane potential. 5 minutes after adding the TRH, we can see that the after hyperpolarization still is there, and then there's an after depolarization. So a depolarization after potential is the term for that, and the bursting is observed during the exposure to the TRH when you look at just an individual neuron. So at 10 minutes post-exposure to the TRH, you can see that actually the neuron is taking on an autorhythmicity very similar to the spontaneously active cardiac myocytes that we talked about. And it is due to this spontaneous depolarization that's occurring after the action potential. So what it's doing is the neuron itself is now increasing its membrane potential close to, if not exceeding threshold, and thereby either driving its own action potentials, as in the latter case, if it exceeds threshold, or allowing inputs to generate the bursting effect. It probably is the spontaneous bursting, because at 16 minutes we see that it just continues to fire, and the after depolarization, or the depolarizing after potential, still persists. I was just checking to see if the textbook contributed any more insight on how this might be occurring. This could be possibly calcium influx, but also could be sodium channels affected, so that we now have a leaky sodium producing this spontaneous depolarization here, this depolarizing after potential. Indeed, in the cardiac myocytes, it's a particular type of leaky sodium channel that wasn't on our list that contributes its autorhythmicity. So this could be sodium, increasing sodium influx, could be inhibiting potassium efflux, but that might be less likely, and could be impacting calcium influx, or maybe a mixture of them, to bring about this depolarization after an action potential. So the effects of the neuropeptides are very complex, and they will be impacting the permeability of ions in many, many cases, especially when it comes to effects on the membrane potential. We can't really change membrane potential unless we alter permeability to ions, which thereby alters the distribution of charges across the membrane. And remember that when you're recording membrane potential, it's inside relative to outside. So if potassium goes out with no other positive ion coming in, then you become more negative inside than outside, so you hyperpolarize. If sodium comes in, you become more positive inside than outside, and you're depolarizing. Potassium accumulates inside, you become more positive inside than outside, and so on and so forth. If you get chloride influx, you get more negative inside, you hyperpolarize. So this is the way that I keep myself on track in remembering what the impact of changing permeability would be, and on the effects on the membrane potential, Are you hyperpolarizing or depolarizing? There are other ways of going about that, obviously, but that's the one that works best for me. So if I see a pattern response, I can almost predict what might be the target of the mediator, in this case peptides. So the co-transmitter peptides, sorry about the misspelling, actions of the neuropeptides are really complex. So you can have a combination of a fast neural response followed by a later slow response, as we were talking about. We saw that pattern of release, and with the TRH even, we saw it taking up to 15 minutes or more to produce the persistent and most profound effect on the respiratory neurons in the medulla oblongata. Now, and that wasn't even co-transmitter interaction, so TRH and substance P can be used as examples to talk about this complexity of the co-transmitter interaction, especially when they're co-localized with serotonin in the medulla oblongata and the spinal cord. So both of these neuropeptides are co-released with the serotonin under appropriate stimulus conditions to the neuron releasing them. The TRH will enhance the response of the postsynaptic cell to the serotonin by increasing second messenger production, while substance P enhances the presynaptic release of the serotonin itself. So a postsynaptic, and a presynaptic effect as examples to that general model that we had looked at in the prior lecture in figure 12.1. Steroid hormones, in addition, can contribute and interact with the neuropeptides to regulate second messenger activity. So this metabotropic effect gives us a very intricate and complex system to produce these types of modulatory responses that we are seeing in the targeted cells. Well, let's take a look at a specific example of this as we begin to consider the co-transmitter effect on the primary transmitter. So this is figure 12.5, where we're considering the interaction of the primary neurotransmitter and a neuropeptide co-transmitter. So one potential mechanism that they can interact, such as vasoactive intestinal peptide, or VIP, and a neurotransmitter such as noradrenaline, is that you can have an increase in the total amount of second messenger within the target cell. So in this case, in panel A, we're seeing the classical transmitter alone with the vesicular release and the synaptic concentration determining the frequency or the likelihood of the receptor being ligated. The greater the frequency or probability of the receptor protein to be in the ligated state increases the likelihood that you're going to have adenylate cyclase activity enhanced. Adenylate cyclase converts ATP, of course, to cyclic AMP that through a cascade activates protein kinases. Kinase means to phosphorylate. So an enzyme, it doesn't mean that. It indicates that phosphorylation will occur, and kinases will phosphorylate other proteins, such as membrane ion channels that we've been discussing, and alter the ion fluxes across the membrane, and thereby contribute to changes in the membrane polarity, or the membrane potential, as we had been discussing. So now if we add a co-transmitter such as noradrenaline with VIP, then we see that the VIP receptor, this diamond-shaped molecule figure is illustrating the VIP, or diamond if you tilt it that way, square I guess if you tilt it that way. Anyway, when the VIP ligates its receptor, it enhances or interacts with the primary transmitter to increase the adenylate cyclase activity so that more cyclic AMP is produced and greater ion redistributions occur, so a larger impact on the postsynaptic target cell membrane potential. Panel C is showing us graphically the increase in cyclic AMP on the y-axis from 100 to 700 in units of picomoles per milligram of protein. So the tissue is harvested after the treatments, as we in vitro, most likely. I was going to say as we saw in panels A and B, however that's highly illustrative only. So you have the same neural tissue where you're proposing that these events are occurring, and you add micromolar concentrations to the bath, either VIP 1 micromolar or noradrenaline 10 micromolar, or the VIP at 1 micromolar and noradrenaline at 10 micromolar together, and you then harvest the tissue and measure the amount of cyclic AMP content reflecting the synthesis of new cyclic AMP. So with VIP alone, you get about 200 picomoles per milligram of protein. With the noradrenaline at 10 micromolar, you get about 100 picomoles per milligram of protein of cyclic AMP. So these are static levels, but that's fine. It shows us a response to a stimulus. And when you combine the two, you get a magnitude of effect, 600 picomoles per milligram of protein, that is greater than if you just added the 200 and the 100. It's actually twice as much. So it's this is called a potentiation, a synergistic potentiated effect, where the magnitude of the response is greater than the total sum of the two separate responses. If these two values added up to exactly what this one would be, 300, then that would be considered an additive effect. If they are potentiating, synergistic potentiating, then it's a more complex system that is occurring, and of course with enzyme reactions it's possible to envision a greater than an additive effect. Now let's consider neuropeptide regulation of protein synthesis after we've completed our considerations of receptor-mediated impacts on membrane potential, the activity of neurons. If you change the activity of a neuron, you're going to change the release rate. So if you release substances, chemical messengers, you have to replace them. So increase and decrease in synthesis and release should parallel each other. I don't know of many examples where you get an increased synthesis and a decreased release, except for in the pituitary gland in preparation for ovulation in female estrus and menstrual cycles. But for neurons, it's not all that common for it to occur. So the hypophysiotropic hormones is a starting point, and we can see that there is a stimulation of synthesis and release that occur for the adenohypophyseal hormones. We'll use this as an example first, but we can envision what would be happening within neurons as well. So here then we begin to look at figure 12-17, and things begin to get a little bit complex with interpreting this figure, but that's our task at hand, is to develop that skill. So let's go through the legend here first. The stimulation of ACTH synthesis and release by corticotropin-releasing hormone is mediated by the cyclic AMP second messenger system. So if we apply synthetic CRH to a culture of rat corticotroph cells, there will be an increase in ACTH, cyclic AMP, and the POMC, proopiomelanocortin messenger RNA, and ACTH release, shown in panels A, B, C, and D, respectively. Let's go and take a look at each of these first and evaluate them. So first we notice on the x-axis it is a bar column, so it is treatments. It's a bar graph with individual columns representing the values for treatments. We have control, and then we've got 2 doses of corticotropin-releasing hormone. The dependent, so those are the independent variables that we can change. The dependent variable in this case is ACTH released into the medium, and the units are nanograms per dish. So control levels are at about 2 nanograms, and there is not a dose response, at least for these 2 concentrations of the synthetic CRH, producing each somewhere around 8 or 9 nanograms per dish. So this shows that the appropriate hypothalamic hormone does increase the release of ACTH, the functional, biologically active ACTH is suspected. It depends upon the method used to measure this. It may have just been using antibodies, so the antibody binding capability of the ACTH is all that is examined. You'd have to test to see whether it was functional. But anyway, we've demonstrated that the CRH causes a release, no difference between the two doses. If we suspect that the cyclic AMP might be the second messenger system, and we take a look at that, measuring cyclic AMP released, the nanogram per dish per hour are the units here, and I'm sorry, it's per hour as well for the units on the x-axis for the ACTH release as well. So the interesting thing about this is that the cyclic AMP is being measured in the medium, not by harvesting the cells. So somehow the cyclic AMP, I guess, is escaping from the cells, and you can measure it in the medium. Anyway, we see the control levels are quite low, but we do see a dose response in cyclic AMP between the two different concentrations of the CRH, the 1 and the 10 nanomolar. So what does this mean to have a dose response in the cyclic AMP levels, but no dose response revealed in the release rate? So that has to make one think a little bit about the coupling of cyclic AMP concentrations to the release. Then in panel C, we are wondering about the effects on the gene expression. So the x-axis here for two treatments, control and cyclic AMP. So cyclic AMP is going to be added in the bath exogenously. So the POMC messenger RNA levels as measured as percent of control. So control, of course, is 100%. And then after adding cyclic AMP, the POMC messenger RNA levels are at about 220 or 225%. So a doubling, a twofold increase with reference to control. Recognize what's happening here. Here we're showing CRH causes ACTH release. Here we're showing CRH causes increase in cyclic AMP. But this, when we compare these two, there's nothing common between these two other than the CRH to say that ACTH release is dependent upon cyclic AMP. These are two independent measurements that don't have any impact on each other. So we cannot conclude from this study that CRH-mediated ACTH release is dependent upon increases in intracellular cyclic AMP. They occur together, but we have not shown that there is a causal association between either of these. And we get even one step farther away from asking the question if CRH is responsible for increasing POMC expression. Certainly it's cyclic AMP related, but if you block cyclic AMP, well, you're adding the cyclic AMP here, so you can say that it is affected by and dependent upon the cyclic AMP. Then finally, we're asking the question if cyclic AMP can cause the release of ACTH. So indeed, the study does make that full turn around the corner and demonstrate that if you add a cyclic AMP to these pituitary cells, you get an increased release of ACTH, here shown as nanograms per 1 times 10 to the 6 cells per 48 hours. So I'm sorry, this is not ACTH released. Well, it is released. But then why are the units different than up here? See, this is per cells. So that must just be a way of normalizing the data, that you have the same number of cells for which you're measuring an amount of ACTH. So if you had 10 times the number of cells in this well as you had in this well, for example, would it produce the same response? Because you just have more cells to respond to the cyclic AMP. So it is ACTH release, and it is based upon the number of cells that could be contributing that amount. So indirectly, then, we can conclude from this that the CRH is acting through cyclic AMP to impact expression and release. This is not the most conclusive evidence because we haven't blocked the cyclic AMP in the presence of CRH to show that it prevents this and prevents this. So how might this be occurring from the extracellular fluid compartment getting all the way to the gene? So this illustration is showing metabotropic types of receptor mechanisms for water-soluble molecules such as hormones or peptides that are the first messengers in the extracellular fluid, and they're coupled to G proteins, relay proteins that depend upon GTP binding to kick off GDP. When the GTP is bound due to ligation, you get a conformation change, reduces the affinity for GDP so it pops off, increases the affinity for GTP so it jumps on. When it does that, reduces the affinity between these two, and now the relay protein can kind of float around in the inner side of the membrane, and then its affinity for the effector increases when it interacts with the effector, in this case adenylate cyclase. It changes the conformation, increasing enzyme activity, and you get more ATP converted to cyclic AMP, as we had talked about before. There can be convergence of different receptors on the same effector, some stimulatory, so this would be G sub S, or G sub I, bringing about the inhibition. Just the same, let's follow it through. The cyclic AMP inside the cell has one mechanism of action. It acts on inactive protein kinase. It's actually protein kinase A, the A for cyclic AMP. Protein kinase A then becomes activated and triggers responses of the target cell, activates enzymes, stimulates cellular secretion, opens ion channels, as we have been alluding to in all the slides previously. But now we ask the question, well, how do we get to the gene expression? So one of the proteins that is phosphorylated by the protein kinase A is cyclic AMP response element binding protein, as shown down here. Cyclic AMP response element binding protein. It is one of many transcription factors that exist and that we'll be discussing. Once phosphorylated, the Krebs has access to the nucleus and can bind to the cyclic AMP response or regulatory element. In the case that we were just looking at, we are looking at the POMC gene. So its CRE would bind the phosphorylated Krebs after CRH had increased its phosphorylation through the metabotropic mechanism we described. And it would make it more likely that RNA transcriptase, RNA polymerase, sorry, I got the wrong enzyme name there, would bind and express the POMC gene, and then it would undergo all the transcriptional and translation and post-translational changes that we've discussed in an earlier lecture to end up providing the ACTH that was measured in the study we just talked about. So it's through transcription factors that metabotropic effects of peptide receptors, peptides through their receptors, can impact gene expression or the synthesis of neurohormones, or whatever the gene product might be. It could be a synthetic enzyme for a neurotransmitter. In addition, as we said, the neuropeptides can modulate neurotransmitter release from the presynaptic neurons. So the release of the neurotransmitter or the neuropeptide from the presynaptic cells, usually through regulating the calcium and the potassium channels. A lot of convergence on these two guys here, as we're seeing. However, in the axon terminals. So an example is CCK regulating dopamine release from neurons in the nucleus accumbens in the rat. This is a limbic nucleus involved in reward and motivation and emotion expression. Neuropeptide Y inhibits the release of norepinephrine in the sympathetic nervous system, so impacting fight or flight type of events. So I'm going to stop here on this slide as we're over 1 hour of recording and complete the rest of this lecture in a